Hi, everybody. Welcome to COVID-19 Q&A with Dr. Daniel Griffin. I'm Vincent Racaniello, and joining me here at the incubator in New York City, Daniel Griffin. Hey,
Hello.
Daniel.
Hello, everyone.
Hello, everyone. And we just recorded a clinical update, and now we are here to take your questions. So let's jump in. Here we go. Oops, I haven't got the right. Let me get the right thing up there. There you go. All right, my third shot was normal. 2 days of side effects on day 5 to 11. Those symptoms returned, unlike the first or second. Anecdotally, hearing others are getting this. Are you seeing this clinically at all?
Yeah, so this is, I think, nice. We're starting to get experience here. We've talked about what was the reactogenicity with the third shot, but remember, those were the first people that signed up, the people who had immune issues, were elderly. Now we're starting to see what happens in younger individuals. And we are actually, I think, as you're, the anecdotes are starting to accumulate. Eventually they'll be processed into data. But for most people, that third shot is ending up being pretty similar to what they saw after that second shot. And that'll come up, I think, when people have questions. If they had an issue with that second shot, what might be some strategies?
I'm trying to adjust myself here because I'm much shorter than you. All right. All I could find in the trials was that 12% of those getting Moderna experienced that reactogenicity for a week or longer. Okay. Scott says, is it a bad idea to get a COVID vaccine if you've had Stevens-Johnson syndrome?
Yeah, so for our listeners, Stevens-Johnson syndrome is, we'll call it a severe immunological response, often to medications, but it can be to something else. And it can go from sort of shedding of the skin falling off, a horrible rash. Stevens-Johnson's actually starts to involve mucous membrane, so your eyes, your mouth. So this is a severe reaction. There was a nice study that just came out this week where they were actually looking at, it was a large healthcare system, tens of thousands of individuals. A number of them had had severe issues, allergic issues in the past, and they actually found that they were able to safely, successfully vaccinate just about all these individuals. But someone who's had a reaction like this isn't someone who should be going to one of these mass vaccination sites. This is an individual who should be talking with their physician, talking to a specialist, understanding what triggered that Stevens-Johnson syndrome, and getting direction on which vaccine would be the safest for them.
Okay. Matt writes, to assuage fears regarding syncytia formation by vaccine spike, are there any specific studies showing that the 2-proline change renders syncytia formation impossible?
Vince, I'm going to let you take that.
I'm not aware of any, but it was my understanding that the 2 prolines lock the spike in a pre-fusion conformation. So therefore, it's not going to undergo fusion. So those vaccines that have 2 prolines shouldn't do that.
Yeah, I'm also trying to figure out how just spike protein by itself not being part of a virus are going to lead to all these sort of fearful things that are being disseminated out there. I mean, if anything, I think we can say billions of people have been successfully, safely vaccinated. These things are not 100% safe, but we now know what the risks are, what the relative incidences of those are.
So that's a good point, because spike alone is not going to fuse because it usually is anchored to a membrane and then it triggers fusion. So I don't think the soluble spike, but maybe the membrane bound in a cell could do that, right? But it's not going to circulate to your heart then and cause fusion. So I don't think those concerns are founded at all.
Yep.
Okay, Darius from Croatia. My question is, are people who had tonsillectomy prone to more severe COVID, or is it maybe the other way around?
Yeah, I don't know if I've seen really good data either way on that. Older individuals, right? A lot of them had their tonsils and adenoids out, but I don't think I've seen any really good data either way on a correlation.
Okay, Leia writes, using antibody tests to detect N protein as a way of distinguishing vaxxed person has been infected. How accurate are such tests? How accessible?
Yes, this is a great question, Leah. We talked a little bit on the This Week in Virology COVID clinical update that will drop this Saturday about the FDA comments about serology tests. And this is the one situation where the FDA actually talks about doing serology tests. The vaccines that we're using here in the U.S. and many parts of the world, so it's important to know which vaccines, the J&J, the mRNA-based ones, are only exposing you to spike. So if a person has antibodies against nucleocapsid, that's going to be evidence that they're having that from an actual SARS-CoV-2 infection, not from the vaccine. So we can actually use these, much like we do with things like hepatitis B, to distinguish between a vaccine reaction and an infection reaction.
And Leah also says, is there a certain time period for testing? Would they be able to detect antibody for many years after infection?
Yeah, so the antibodies to the nucleocapsid, the immunodominant protein, are not being generated in all individuals. We've actually seen the milder disease. We've seen a higher percentage of people that remain serology negative, the more severe. But what is the durability of those antibodies? Time's going to tell. So will they continue to stay positive for years? I don't know.
Stephen writes, since the J&J vaccine is DNA-based, does it reside in the nucleus longer than the mRNA vaccine in the cytoplasm? Does that affect the making of spike proteins?
You know, we think of this as sort of a similar sticky note, a short kinetics. This isn't something that's, sitting there at high levels in your nucleus for long periods of time. But this is, you're bringing up the distinction. The mRNA vaccines only need to get into the cytoplasm, and then we're doing the work. The DNA-based, the J&J vaccine, adenovector, so that'd be the AstraZeneca as well, is going to get into the nucleus, and that's the way it gets produced.
Okay, Sachs Girl writes, any new insights, advice regarding COVID-19 and type 2 diabetes?
So we clearly know that individuals who have diabetes are at significantly increased risk. So having diabetes alone is going to put you at that 10% risk for ending up in hospital if you get infected with COVID-19. So this is certainly a group. What is the best thing you can do is get vaccinated, get a full vaccine series, and then all the other things that we can do. We're still in the middle of a pandemic, so think about all those decisions about safer choices to reduce your risks.
Daniel, people are asking me to put a Post-it note on the blemish on the wall behind me because they keep rubbing their screen. Zachary writes, maybe I'm crazy, but did you see the Times article today where they quoted the cardiologist who said masks are our best mode to stop transmission and we need them forever?
Zachary, you are not crazy, but that cardiologist might be. Vincent and I were talking about that. There's sort of reasonable expectations and there's unreasonable expectations. If you look at some areas of the world like Denmark, like Portugal, if you can get to a point where you have very high vaccination rates, you can actually go back to a different way of approaching this. I do not see a world in which we have to wear masks always everywhere. So I see an end to that.
Daniel, what do cardiologists specialize in? Infectious diseases?
I think so. I think cardio, it's a type of the cardiobacterium. Yes. No,
okay.
They are heart doctors. Stay in your lane.
Kathy writes, good afternoon from Seattle. Curious what the latest data is on Novavax and when it might come to the U.S.
Yeah, so Vincent, you always give me a hard time when I give these broad confidence intervals. We are hearing that one of the biggest issues was really a production challenge. We want to be sure that Novavax can produce the vaccine in a reliable, in a significant quantity. They're in discussions in the UK. We are in the end of October. We're really hoping November, December, we're hoping this year does not end with Novavax not becoming available. And I really think that this would be important to have another option. The more choices people have, there are certain people out there who would feel more comfortable and who actually are still waiting. I know they're getting tired of waiting, but we don't have any sort of a firm date yet.
Jason wants to know if you can update us on any planned studies for long COVID.
Yeah, I can give you a little bit of an update here. I was staying on a call yesterday with Raul Rabadon, I was talking with him about the NIH is moving forward. They're making snails look fast, but they have given a lot of the money to NYU, and then NYU is going to do sub-grants. NYU is, say, a prestigious medical institution here in New York City. I actually graduated there. But a lot of people have grown tired of waiting, and so a lot of people are jumping up and starting trials. But, yeah, it's been a long process. They want to make sure it's done right, but, boy, it's taken a long time. So when I know more, I will certainly update everybody.
Nick wants to know what's your tie today.
Oh, so today it is Antibody. So I don't know if people can see this.
Wow, look at that. That's good.
It's a crystal structure of
any particular
the light and
antibody?
heavy chains. I think that these target the spike protein, but I'm have to check their affinity for you.
Mary wants to know if we can get a follow-up on the 13-year-old with Downs.
So, Mary, I can give you good news. She actually got out of the hospital, and she's doing quite well. She's actually home with her mother and father, so a happy ending.
Zachary writes, why are we now seeing arguments for boosters every 6 months? Seems absolutely absurd, and it's like these people are convinced this pathogen is magic.
I think today, right, you probably started to see discussions of fourth boosters for certain people. So, I'm not sure about sort of the psychology behind a lot of this, but people are really frightened by this virus. People are really, I think, upset by this pandemic. The reassuring thing, these vaccines continue to do what they were designed to do, but now there's this discussion: can we get more out of these vaccines? Maybe we can get more protection against infection. Time will tell, but there's a lot of interest. But the way this pandemic ends is people getting their first and second doses.
Nova guy writes, why hasn't Moderna for under 18 been approved yet? Didn't they submit for approval in June?
Yeah, there have been some delays there, so I don't know the specific delays.
Mary writes, we hear all sorts of cautions about overinterpreting VAERS. How is it properly used?
Yeah, so the way that we suggest that VAERS should be used is to generate hypotheses and then to look specifically into it. We joke about all the stuff that can be dumped there. I mean, there's even the report of some individual turned into the Hulk, right? Which, it is an open platform. It's designed to be open. You don't take any sort of number from there. You look there and you say, okay, we're hearing reports of a certain thing, and then we look into it with other. But you never take VAERS data and just directly analyze or publish it. It's hypothesis generating. Oh,
ECDSA, is the data on iota carrageenan nasal spray in terms of protection any good, or is it something that's not likely to have any effect?
Yeah, I don't know if I have any good information on that.
Cat Lady says, you're doing awesome. Stay strong. Carry on. I appreciate your work. Thank you very much. Could you guys discuss some of the general highlights of different vaccines? Is one shot better than the others? Do you break it down by patient population?
Sure. Let's go through this. As Vincent and I were talking right before this live stream, the mRNA vaccines, the Moderna and the Pfizer, really are excellent. The J&J with one shot, I mean, we were at 81% protection against hospitalization and death. With the second shot, we're thinking that's going to get up to the same level as we saw with the mRNA vaccines. They presented that data. FDA said, boy, your confidence intervals are pretty wide. You need more numbers to say that confidently. But we're seeing really good persistence of antibodies with J&J. I personally do not think it's an inferior vaccine. But let's look at different age groups. If you have a younger individual, let's say it's a young male, you may have certain decisions about whether or not you want to do an mRNA vaccine. If you have a person who had a bad reaction to an mRNA vaccine, this is why it's nice to have the J&J as another option. I think each different individual is going to be wanting to talk and think through. They're all great vaccines. In a lot of ways, it's, do you like Coke, do you like Pepsi, or are you a fan of Sprite?
Daniel says, I vaxxed in March, got COVID in July. Should I get a booster?
So interesting. We know the other way around, right? We know people who had COVID and then got 2 vaccines. Those people look like they're in a really good situation. We've seen some really good studies on the antibodies in those individuals. But what about you? You sort of did it backwards. You got your vaccines, then you got a booster by getting naturally infected. We don't know for people like you. We do know that you're gonna have really good protection against hospitalization, death. You survived your infection, probably did better than you would have done if you hadn't been vaccinated. We don't really know. And again, when we get back to the boosters, the sky's not falling here. We're just thinking that maybe we can get a little more protection with the boosters.
Christina writes, is a 3rd booster needed for those who have interstitial cystitis?
So again, we don't know, and I'm gonna say that a lot, and hopefully that won't frustrate people. They'll see it for the honesty that it is. You've got to ask what you're trying to get with that booster. If you're a frontline healthcare worker, if you're trying to maybe decrease the risk of getting a positive test and ending up missing work and being unable to be there for our patients, then okay, there might be a good argument for a booster. If you're an older individual who maybe didn't get as robust a response as we were hoping, right? That's the over 65 recommendation. I'm not sure interstitial cystitis is really going to fall into a situation where we're going to feel like you're going to be at higher risk. So I'm not sure a 3rd booster is needed.
Mark writes, could you talk about Fluvoxamine?
All right. Well, we spent a lot of time actually talking about Fluvoxamine on our TWIV clinical update that we just recorded. And there was exciting data. We got results of the Together trial, which is out of Brazil, out of South America. And what is Fluvoxamine? I like to say it's a not so selective serotonin reuptake inhibitor. It's a medicine that we've been using here in the States since last century. So we've been using it for decades to treat people with OCD. But in addition to its effects, it also has an anti-inflammatory effect, and it's been studied in mouse models for quite a while to block sepsis, sort of this exuberant cytokine storm, over-exuberant immune response. That's the way we think it's working. And the Together trial did not show an impact, positive or negative, on viral replication. So we don't think it hamstrings our immune system's ability to respond during that first week. We don't think it has an antiviral effect during that first week. But it does seem to, at least the data in the Together trial, reduce an individual's risk of progressing to ER or hospitalization. And there was encouraging suggestion that it may have a mortality reduction as well. One trial, very exciting. We've got a mechanism, and there's several other large trials that are ongoing.
Mike writes, does chronic systemic scleroderma warrant a COVID-19 vaccine exemption?
Yeah, so this, I think, gets back to one of our first questions. What we're worried about here, right, is an individual with chronic systemic scleroderma. Are they going to have a bad reaction to the vaccine? And what I always put this in context for is how would a person like that do if they got COVID? There was the large healthcare study that I probably should talk about on the next clinical update, where they looked at tens of thousands of healthcare workers in a particular system. We're able to show that these individuals did quite well with vaccination. I would say someone with chronic systemic scleroderma, they're really an individual who would benefit from sitting down with a specialist and discussing the pros and cons and how best to approach this.
I'm 9 months out from a second Moderna jab, on the fence about the booster. Is there an outer time limit as to when a 3rd shot would be too long to wait?
Yeah, so this is an excellent question, Kathy, because some people are worried. What if I wait too long? Will I cease to be eligible? Will it not work? What we understand about immunology is that you generate memory T cells, you generate memory B cells. Waiting longer is not a problem. You're not going to fail to boost. Sometimes I worry about that with the vaccines, where we'll take the shingle shot as an example. They say you should get your booster, 2 to 6 months later, and then it's a little over 6 months, and people are worried, do I have to start all over? You don't have to start all over. There's no reason for us to think you have to start all over if you wait 10, 11, 12, just, trying to make that decision.
John writes, I have a 5-year-old who I'm so excited to get vaxxed but concerned about small cohort size in this study in regards to safety. Is this a concern? Should I wait?
So the big discussion that the FDA Independent Advisory Panel had was exactly what you bring up, is that this is a small cohort. Do we know about rare events? Do we actually know what the myocarditis risk will be using this lower dose in this population? There's modeling, there's extrapolation. Some parents are looking at this, and even just for the mental health of themselves and the children, we anticipate that a third of those 28 million kids are going to get vaccinated shortly. So if people are on the fence, if they're not certain, it's only a matter of weeks before we have more data to really help with that risk benefit.
Jenny wants to know, why should I get a booster that's not effective against variants?
Jenny, excellent question. So the third shot we think is effective against the variant. So let's go into that. A lot of people are saying, I just want to wait till there's a Delta specific or a variant specific. But the interesting thing, and I know the folks on Immune have done a good job of discussing this, is there is an ongoing evolution of our antibody repertoire over time. And when they've looked at it in small numbers, I'll give you, that third shot actually looks like we are developing robust neutralizing antibodies against Delta. So even though the shot was not specifically designed for Delta, the data we're getting is suggesting that that boost is generating antibodies not just against the original variant, but against all the other variants that we've seen so far. Will it work against all the other variants that we may see going forward? Not necessarily. So the companies have been asked, and they're poised to, if they need to put in a different cassette, a different mRNA sequence, that can be done, we think, with a turnaround time of about 100 days is what I've heard.
M. Pepper, breakthrough infections abound with vaccinated people infecting each other where I live, or the unvaccinated children infecting the vaccinated adults. Are you seeing this?
Yeah, so this is something that I guess I'll sort of clarify on. I hate to use the word breakthrough because it's suggesting that that we were expecting vaccines to prevent infection. Vaccines historically, and I always love to use the injectable polio vaccine as an example, vaccines protect us from getting severe illness. They protect us from getting sick. They're a fire extinguisher. They're not body armor. So we certainly see people who have been vaccinated get infected. That is actually at a lower risk, so there is some impact on reducing your risk of even getting infected. Certainly a reduction in your risk of getting a symptomatic infection, certainly a reduction of hospitalization and death. Do people who are vaccinated, do they get infected and spread to others? Yes. We say that's reduced about 50%, and we have some science maybe suggesting why that is. It looks like that period of contagiousness is probably shortened. You might reach the same peak, but it looks like that's shorter. But vaccinated people can spread it to other vaccinated people. That second vaccinated person, again, the same dynamic. It's harder for them to get it, so there's some protection against initial. If they get infected, there's some reduction in that transmission chain going forward. So yeah, vaccinated people can get infected. Vaccinated people can spread it to other vaccinated people. Unvaccinated people are at least twice as good at every one of these steps as the vaccinated. And they're also at that high risk. We have a 2% case fatality rate for COVID still in this country. So 2% of all the confirmed cases have resulted in death. And children, the unvaccinated children, hopefully this is about to change, but I talked about a number recently on the clinical update. Here in the U.S., over 600 children have now died of COVID. So we're hoping to get those unvaccinated children some protection.
BV wants to know how long it takes to build up your bow tie collection.
Many years, BV.
Can obesity by itself, separate from the conditions associated with it, result in worse outcomes from COVID?
Yeah, so this is, Jack, this is a good question. We don't really know if it's something intrinsic to the obesity, but obesity by itself, if you separate all those other things out and you just have a BMI greater than 25, just a BMI greater than 35, you don't have diabetes, you don't have hypertension. The obesity itself is a significant risk factor. This was a significant risk factor with the H1N1 influenza. So there might be something about body fat that figures into the cytokine response, into the morbidity associated with infection.
My cousin has Guillain-Barré. How did her whole nervous system just shut down?
Yeah, so Guillain-Barré is something that we, I'm going to use influenza as an example here. We see it with influenza vaccination, but we see it much more often with influenza natural infection. We saw a lot of Guillain-Barré with acute COVID. Actually, I can think of times we had multiple patients in the hospital with this. It's actually affecting the nerve conduction. So it's really the wires getting damaged. You can think of it that way. But yeah, really a tragic thing. Fortunately, we're not seeing a high rate of this with our vaccines.
Would there be any reason for a person with Polycythemia vera not to get vaccinated?
Polycythemia vera. So this is having lots of red blood cells, right? Sometimes this is, we say secondary because you smoke a lot and it goes up, but some people have primary. They just produce a lot of red blood cells. There is no reason for a person like this not to get vaccinated, but this might get into the subtleties. What vaccine would I recommend in an individual like this? Probably an mRNA-based, because the vaccine-induced thrombocytopenia, thrombocytosis, VIT, make it easy on myself. I just would worry about that, even though that is incredibly rare. We know those numbers are low. This might be a person that I say should get vaccinated. I would recommend a Moderna or a Pfizer shot if you were here in the U.S.
David writes, are you giving monoclonal antibody infusion for prophylaxis after exposure? If not, any idea when this will be available?
So we are certainly doing this. I think I discussed, I don't know if it was this or where I had a gentleman in the hospital. So he was in the hospital, got exposed to someone who was positive. We ended up giving him this prophylaxis after exposure. He did turn positive about 4 days later, but he was completely asymptomatic. And this is a gentleman with a tremendous number of medical problems who would have been high risk otherwise. So we are giving monoclonal antibody infusions for prophylaxis. There's also a number of individuals with ongoing exposure. I have a pediatrician who is unable to respond to the vaccines. Actually, this was a really horrible reaction after his first vaccine dose. So he's getting that repeated dosing, as I've talked about. But yeah, this is available. This is part of the REGEN-COV EUA.
So people are saying you're low, so I'm going to boost the volume here. So give me a sec to get into the menu. Daniel is input number 2.
And I'll even move it closer. Let's get a good...
And
he likes to move back away. Stay. All right. Can people live tell me if the volume is any better? Is Vincent doing a good job?
yeah, it's I boosted it up a bit and you brought it closer. So let's see here.
Hopefully we are good.
All right. Daniel, in terms of immunity to transmission, shouldn't we be looking at this like immunity to OC43 or other common cold coronas? OC43 transmits just fine in the face of population-level immunity.
Yeah, OC43 is an interesting one, right? I like the mnemonic I use is old cow 43. This is one of the ones where we think when it first entered the population, there may have actually been something of a high mortality and a lot of disease when it first came in. But we do think that now this is sort of part of the common corona background, people getting it when they're younger. But yeah. OC43 transmits just fine. We've seen the modeling, and we saw the Jeff Shaman data where if someone gets OC43, they can get OC43 again. Infection doesn't protect you forever. Maybe it's about a 9-month, and then after that, your risk of infection goes up. So again, we're back to what are we trying to do with vaccines? We're trying to keep people from dying. We're trying to keep people out of the hospital. We're trying to keep people from getting all the post-acute sequelae of COVID. Do we think that we're ever going to get rid of SARS-CoV-2, that it will go away? I don't think so. It's in the deer. It's in the rodents. It is now part of our experience.
So they're saying the volume is good. There you go.
All right. Thank you.
Natural immunity. Any reason to get vaccinated? Which is better, vaccine or natural immunity?
Okay, Victoria. Is the V really stand for Victoria Vaccines? I like the alliteration there. So Victoria V with a vaccine versus natural immunity question. The way I usually answer this is I say the question here is if you have been infected with SARS-CoV-2, if you had COVID, is there any benefit to getting vaccinated? And we have that really nice data from the MMWR where an individual who gets vaccinated after being infected drops their risk in half for getting a second infection, right? So that means, I think about that, people who've been infected get reinfected. People who've been infected and vaccinated, they can get reinfected, but at a lower rate. One shot of the mRNA vaccine was not enough to get us a statistically significant reduction in those second infections. It took 2 shots of the mRNA. So I know there's a lot of people out there talking about antibody levels and saying people only need 1 shot after prior infection. We are recommending full proper vaccination after that first infection so that you don't get a second infection. We anticipate that people who don't get vaccinated will, and it'll be just a question of when they get reinfected that second time. We do not know as much about the vaccines about what that second experience will be like. Will it be less severe? Will it be more severe? We're not sure.
Mike says, what role does blocking complement play in prevention or treatment of COVID?
So, Mike, I don't think we really know enough about the complement system and its role in COVID at this point to say, but this is certainly an area of active research. And part of it is because we do think a lot of the morbidity, a lot of the trouble people have is because of activation of the immune system, and complement is part of that.
What do you think about delaying the second dose to 12 weeks? I plan on this for my 8 and 11-year-old.
Yeah, so that was something that we stumbled into. I mean, there was a lot of passionate debates about what they were doing in parts of the world where they were not giving it 3 to 4 weeks later, waiting for that 12 weeks. It does look like you get a more robust antibody response. T cells are doing just fine after 3 to 4 weeks. So a lot of the ideas of why did we do 3 to 4 weeks, why didn't we give it this 12-week, was that we were in a pandemic. We were able to get into the 90s with only a 3 to 4 week wait. Is there a problem waiting 12 weeks? I don't think so.
Chelsea says, are the mRNA boosters an actual booster or a third dose? Is there a difference?
Only with Moderna. With Pfizer, you're just getting a third shot. With Moderna, you're getting a half dose, right? So the Moderna is 100 micrograms, and the booster is 50. Pfizer's 30, 30, 30. And if you get a fourth dose, it's also 30.
Gail writes, does it make sense for a Pfizer vaccine recipient to get only a 50 microgram Moderna booster without any data showing what benefit? Mix and match study was based on a 100 microgram Moderna booster.
Yes, Gail, that's actually that's very sophisticated, and people have been bringing that up. They authorized Moderna as a 50 microgram third shot, booster shot. But in all those studies that were presented at the Friday FDA meeting, those were all antibody levels, and eventually we're going to get T cell data with a 100 microgram boost. So yeah, when you start looking at the mix and match and they say, oh, we got a little bit higher when 2 Pfizer people got Moderna, remember, they got a little bit higher giving twice the dose that is authorized. I suspect that if you had 2 doses of Pfizer and you want to stick with Team Pfizer, you're going to get a very good response antibody-wise to getting a third dose of Pfizer.
Mark says, could you touch briefly on how you could talk to someone that is hesitant about vaccines? Do you believe there are people whose mind won't be changed?
Well, Mark, I think the most important thing, and the way you talk to someone who's hesitant about the vaccine, I say listen to someone who's hesitant about the vaccine. I think we've done a really bad job of taking people and shaming them, of categorizing them as anti-vaxxers, et cetera. There are some anti-vaxxers. There are people that make a lot of money promoting that. But there's a lot of people that have questions. There's a lot of people that are vaccine hesitant, that are not sure. I don't think as a medical profession, we've always done such a great job of listening. I think it's an average of 6 seconds before we interrupt someone when they come in. We need to do a better job. And what is the big thing? Yeah, if someone's hesitant about the vaccine, listen. Unfortunately, there are mass efforts. We now know Facebook is doing this, shall I dare say, on purpose, of providing misinformation, disinformation. And these people are really often the victims. And they're the victims in 2 ways. One, they're the victims of really a well-orchestrated disinformation campaign. And they're also the victim, too, because they continue to remain vulnerable to this devastating disease.
John writes, if infection generates wider, better immune responses versus vaccine, why are infected and unvaxxed people more likely to be reinfected than infected than vaxxed people?
Yeah, I'm not sure we know that's true. There was a non-peer-reviewed preprint out of Israel that really got circulated a lot. It was absurd numbers. It was like hundreds of times more protected. But that's not what we're seeing. And I've shared like the Oklahoma data, where it actually looks like people who were infected before compared to the vaccinated were actually 2 to 9 times more likely to get reinfected, right? So I don't know if I'm confident saying that prior infection is giving you more protection than vaccination. All
right, so overjoyed is 18 plus months long COVID, reinfected less than 90 days prior to the first shot, and between shots 6 months without infection. Is a third shot okay? Non-durable immunity before vax.
Yeah, so let's see here. So 18 months, long-haul COVID, reinfected less than 90 days prior to first shot between vax. I think the third shot is okay. What we're actually, and I'm trying to get feedback from individuals on, is individuals like you, overjoyed, who have long COVID, who got a couple shots. How are they responding to that third vaccine? I have not heard any concerning reactions.
Kathleen says, can you please ask him about fog brain? Well, you could ask him.
Kathleen, hello, Kathleen P. I talked about a paper, so listen to the clinical update that comes out this Saturday. But we've begun, I think, to realize that the COVID fog brain, the brain fog, is more than just brain fog. This is actually true cognitive dysfunction. So there was a nice study that I went through where they did objective testing. It was out of the group at Mount Sinai. Got some excellent folks over there. And these people have problems with memory. They have problem with executive functioning. Why? We do not know the mechanism yet. And that's the whole idea behind all these post-acute sequelae of COVID studies, to try to get at these. But in their group, it was young individuals, median age, I think it was about 49. It's slightly more female population than male over there at Mount Sinai. But these are individuals who actually have real cognitive deficits, ongoing real cognitive deficits for months and months. And we still don't really understand the mechanism.
Is there any understanding of the early disease process that is causing anosmia? We know that olfactory sensory neurons don't express ACE2, so is this the case of inflammation?
Yeah, so we don't know exactly. There is some early work that it's really the sustenacular, so the supporting cells that are being affected by acute COVID. But yeah, we don't, and I'll say going on, we don't really understand about the late process. So certain people will respond, will get better after a short period of time. But unfortunately, a lot of individuals continue to have this as a long-term impact. Some of those people will start to get smell back. It's interesting. Usually the process, they start to get weird smells. Things smell off and metallic, and then the smell. So when I start hearing that, for me, it's encouraging that they're starting to get smell function back.
Louisa, right? Is there a health risk for a 4.5-year-old to get the 5 to 12-year-old shot?
So the reason in part why they pick the certain ages, right, they're not completely arbitrary, is there is an age and not a weight-based development process for the immune system, right? So I know a lot of people are, saying, my 4.5-year-old is big for his age. Well, his immune system is that of a 4.5-year-old. So, we're getting pretty close. I understand how frustrating it must be if they approve it down to 5 and you've got someone who's 4.5 years old. But the EUA is pretty strict. So that child is not eligible yet, and we'll have to get more data. Hopefully we're just talking about a few months, but yeah, it's been a long wait, and I'm sorry about that.
Nick writes, any thoughts or data on whether someone with myopericarditis should avoid getting mRNA vaccines as a booster?
Yeah, so I'm going to break this down into the two issues. So the myopericarditis, if this is an individual who had that, and we'll say completely separate, right? That was something that was in the past. We do worry because we know the incidence of this with SARS-CoV-2 is actually quite high, much higher than we're seeing with any of the vaccines. And this came up a lot when they were talking about moving the age down. But what about a person who got an mRNA vaccine? Let's say they got the second Moderna shot, and then they develop myocarditis. That would be an individual that we would probably talk about switching platforms, but also be a person you want to have a real heart-to-heart. What are we trying to accomplish with that booster? Is the risk-benefit there for them? So this is actually kind of one of the questions that I was hoping people would get at. Not everyone needs to rush out and get a booster. If you had a bad reaction with one of those first or second vaccines, you want to be having a discussion about what the goals are with further boosting might be.
Claire writes, I'm 70, had 2 Modernas, and wonder which vaccine choice for a booster is best.
So they're all fine. There's no bad choice here. Claire, I'm in the same boat. I got 2 Moderna vaccines. I may get a J&J as a third dose, but we're guessing. We don't have any really good comparative head-to-head data on efficacy because that's what we care about. We don't care about how high those antibody levels get. I mean, I know that's the competition. We're not sure what measuring those memory T cells, circulating memory T cells are telling us. What we're really gonna find out over time is the efficacy. If you got 2 Moderna vaccines, if you tolerated them fine, if you want to go ahead and get a third Moderna vaccine, I think that's a fine choice. If you want to jump over to the dark side to get one of those Pfizer shots, that's fine as well. And J&J, there's a lot of people suggesting that might be a fine choice, but we're all guessing. We don't have the data. We're stepping beyond the science here.
Any insight on when older adults in long-term care can get their booster if they've recently received monoclonals? Sure.
So we have guidelines on this where we recommend waiting 90 days after getting the monoclonals before we do the booster. Now, this really was extrapolating from using vaccines that used attenuated reproductive competent virus. We're not really sure that monoclonals are going to impact your response to an mRNA or even an adenovirus vaccine. So we do make that recommendation, but there are case studies already out there showing where the vaccination has happened sooner, and we're still seeing robust antibody and immune responses. So the current recommendation is wait 90 days and then go ahead. But as the science evolves, we may remove that waiting period.
Can I expect more side effects if I get both the flu and a third dose Pfizer shot, one in each arm?
I wouldn't expect very much. We're not seeing any sort of synergy in the reactogenicity. So if you get a flu shot and you feel a little bit from that, if you get your third Pfizer shot and feel a little bit from that, there might be a little bit of the additive, both arms hurt, but no.
What is it about being diabetic that puts someone at higher risk of being hospitalized? Something that can be controlled like glucose level or insulin or something else?
Yeah, it's something else, Ralph. We don't really understand completely. But diabetes is not just about having issues with your sugar. There is an immune disturbance in people that have diabetes. They're at more risk of a lot of infections, bacterial infections. Here, they're at more risk of worse outcomes with viral. Even diabetes, even a diabetic who has really controlled sugars is at higher risk. Boy, a diabetic who has poorly controlled sugars is at even higher risk.
They say that the kids' vaccine is a third of the strength of adult. What does this mean? Does it matter?
Yeah, so this is the amount of the mRNA. So the mRNA in the Pfizer that we're using for the 12 and up is 30 micrograms. In the pediatric, so the 5 to 11, it's only 10 micrograms. Now, the nice thing is they've actually looked at the amount of antibody levels that are generated with this, and we do know on a population level there's correlation there, not in an individual, and they are actually getting the same levels that they're seeing in the adults with this smaller dose. Kids really, kids are good at learning. I think we all know this, and our immune system is much like the rest of us.
Let's see here. 68 years, Pfizer times 2, 8 years post-stage 2 LCIS with radiation, high EBV titer, prone to bronchitis, depression, fatigue, booster, J&J, Moderna. Can I get full Moderna?
That's interesting. Yeah, no, so what you bring up is exactly what was presented the other day. So people who got 2 doses of Pfizer, and if you're all about antibodies, you're going to get that slightly higher level with the full dose Moderna. That's what they were presenting, not the 50, not the half dose of Moderna. The way it's authorized in the U.S., it's authorized as the 50 microgram Moderna booster. Plenty of people, though, are just going into pharmacies and getting the 100 microgram dose.
How soon do you regain full taste smell? I've been hyposmic for 7 to 8 weeks with fluctuations.
Yeah, this is frustrating. I've definitely had a lot of patients who have had really long-term issues with smell. And I think, sort of this, it's not completely gone, but it's diminished. It's not that good. I have a patient that I was speaking to this week, and he didn't even notice, but everyone was like, why are you putting so much salt and spice in all your cooking? And he had, this decreased ability to taste and smell. We don't have any great therapeutics. We've tried doing like the steroid nasal sprays. We're not really sure that that's speeding things up. It's just time. And unfortunately, for some individuals, this can go on for a very long period of time.
Role of iron status. Is a person with low ferritin less likely to develop immune responses? Risk of adverse side effects. Could free iron from supplements elevate cell damage?
Yeah, so this is a great question, Andrea, and it reminds me of a discussion, Vincent, we had with Dixon talking about iron supplementation in malaria, where people have this idea. A lot of individuals are iron deficient. We got to supplement them, get them up, and they actually had worse outcomes. They had an increase in mortality because a lot of microbes really like iron. I don't think we have any compelling evidence in COVID that iron supplementation is going to do any harm, but I don't think we've specifically looked at this with the granularity to know for certain.
Huge fan of your updates. I have the opportunity to take the 3rd dose of Pfizer 8 months after the 2nd. I'm a healthy male. Is it safe to get the shot?
Yeah, it's safe. We're again, you're healthy. I'm looking at your picture. You look like a healthy 36-year-old male. There's no safety concerns. That's one thing that we can say.
Do you guys notice how nice Daniel is compared to me? He's got a nice demeanor, bedside manner. Boy, that's doctor training, right? That's great. And also personality, too. My 2 kids, age 5 and 8, had COVID for the first time in August. I'm a hospital nurse. I'm a bit lost from coworkers about if I should get the girls vaccinated once approved. Any advice?
Yeah. So, you don't have to rush, right? Remember, you're going to have a little time. There's about a third of the kids that parents are just like chomping at the bit. When is that first day? When can I get them in? But here you run into this issue that we've talked about. If your children got COVID and you say it here, the first time, we anticipate without vaccine, there's going to be a second time, maybe even a third time. It's not clear that children only get infected once and don't have repeat infections. We've certainly seen repeat infections in this age group. So we do encourage that. And the encouragement is that individuals, your 2 girls in this example who had COVID, we would encourage them to get vaccinated to have that extra layer of protection. The large study we have in adults, you're going to drop that risk by more than half. Well, boy, if people keep getting boosters over time, it may get even smaller and smaller. But yeah, don't feel like you have to rush into this. Think about it. If you're a little bit concerned, wait and see what's going on. But yeah, in general, we would recommend vaccine.
Is it appropriate to do immunity tests before doing a booster?
Peace and serenity. Thank you for asking that question. No. I get this call multiple times a day, and they always want to start the conversation with, I check the antibodies, and then it goes from there. We do not know on an individual level what to do with serology testing. And the FDA has, I think their warning is as strong here as it is like against using horse paste. You're not supposed to be using the serology test to make clinical decisions because on an individual level, we really don't know what that means. When we looked at individuals that have gotten infected, individuals with very high levels have gotten infected. Individuals with low levels have not gotten infected. So even if your antibody level is high, we don't know what that translates into as far as risk for you as an individual. So no reason to check those. Just look at the risk benefit of your age group, your other morbidities, and then make that decision about whether or not you want to go ahead with a booster.
You think it's worth getting a booster for somebody who is intermittently on methotrexate?
So that would be one of the people that fell into the early category. So methotrexate is a medicine that suppresses the immune system. We use it in a lot of rheumatoid arthritis, psoriasis, a lot of other diseases. And we would expect this to interfere with the immune system. So this is one of the people that were in the first tier of vaccines being recommended for.
Daniel, thank you for your contribution. Really appreciate it. Really appreciate your support. A friend of mine has Berger's disease. Condition got worse after getting vaxxed. He won't get COVID vaccine. Could monoclonals be an alternative?
Yeah, so this is something that I've tried to bring up repeatedly. We have individuals who had maybe a bad reaction to that first dose of vaccine, and some of them are pretty severe. And so this person then, the last thing you want to have them do is get COVID. So this is a situation where that post-exposure, ongoing exposure prophylaxis can be considered. Right now in the United States, it's Regen-Cov that only has the EUA expansion to cover that, but this would be someone who should be considered for that.
Does a fully vaccinated church choir safe enough to return to unmasked singing?
Okay. So, Barry Lou, you got to put this in context, right? People who are vaccinated, they themselves are at lower risk of severe disease, but they still can get infected. They still can transmit. So you've got to look at who's involved and decide how you're gonna take that risk. Ideally, you're outdoors. Ideally, there's good ventilation. But you've got to put this all. A vaccine is not a body armor. It's a fire extinguisher. It helps put things out, but there still can be transmission and infection.
Since there are more than a few research papers on how aspirating the needle prior to vaccinations mitigates myocarditis and thrombocytopenia, is this being considered at the clinical level?
Yeah, so not yet, and I think this is interesting. I mean, we brought this up early on. I'm old. I was originally trained that when you put in the needle, you pull back on the plunger, and there is some concern that maybe the amount of vaccine getting into the vasculature is somehow related here, but I don't think that's gotten to the level here where they've actually made recommendations.
So related, Lillian says, should we ask for aspiration before getting vaccinated?
I don't think it's bad. I mean, the reason we stopped doing it is it does increase the pain. There's actually a discomfort when you pull back, but it is pretty minor. Maybe just find an old doctor like myself, and we have it so trained into us that it's hard for us to break that habit. But I don't think there's a negative to doing this.
David, thank you for your contribution. Let's see. Is there a higher risk for low-age kids to get bad, sick COVID, and any research for vaccines as safety and efficacy in this group?
Yeah, so a lot of the discussion by the FDA Independent Advisory Panel the other day was really this sort of risk-benefit. So what are the different risks to children, and what are the risks associated with the vaccines? And it was a really small group, right? So they're extrapolating the risks as they goes down because we do not have hundreds of thousands. We don't have millions of these younger kids getting vaccinated. So they actually compared the risk of myocarditis with COVID and more severe myocarditis that develops with COVID to the less severe and less common myocarditis. They looked at the fact that some kids might have an adverse reaction to the vaccine, might end up in hospital. Again, a very rare event, but comparing that to about the 1% hospitalization rate we're seeing in this age group in the United States. So that was a lot of the discussion there. And so the way the panel came out with the recommendation here, is that we think this should be made available to parents who want to vaccinate their children. We don't want to stand between them and access to the vaccine. And then as more data comes out, we can have more comments.
I know you've touched on this before. What are your thoughts on masking in schools?
Yeah, so I think your second, sort of carry on, is there a community transmission level when it's safe to not do this? Is there a vaccination level when it's safe to not do this, et cetera? I don't think that we're going to be masking forever. I know there was a New York Times piece suggesting that we'd be wearing masks for the rest of our lives. But no, there is an end point here. When it gets below a certain level, there's actually a program that the CDC is piloting, this test to stay. If there's a way to keep the COVID from transmitting in our schools, it would be great to get those masks off the kids. We're not quite there yet, and I don't think we have any firm metrics yet on what that number should be. But yeah, this is something people are actively working on because it's not great having to wear a mask.
Here's one, Daniel. Why isn't the Michael Mina protocol being implemented?
It should be. It's actually, I have to say, it was interesting here in the U.S. In a lot of parts of the world, there's really good access to rapid testing. But I think we're fighting an uphill battle here. Once you come to believe something, it's hard to change your mind. And somehow people here in the U.S. got really focused on PCR as the gold standard and started comparing the antigen test to PCR. The antigen tests are really good at identifying people who are infectious. You are going to pick up too many unnecessary people with PCR, and I don't think we've sort of appreciated the harm we've done with picking up people who are not infectious and locking them away for 10 days. But yeah, there is a tide change. We're getting there in the future, but boy, it's going slowly.
Monica, thank you very much for your contribution. Those of you who would like to support us, there's a little button down below. You could contribute. And if you don't want to do that, you could just give us a thumbs up. We have over almost 600 people here in the chat, and you could just give us a thumbs up. That would be great. Let's see here. You once said that the chance of dying from COVID after being vaccinated was 1 in 500,000. Is this still true?
Yeah, actually, I think that this actually is affected by, I'm going to say a couple things, by the population you're talking about. There may even be a time after vaccination that fits in here. So one of the things I comment here in the U.S. is we've seen about 750,000 deaths from COVID. We've seen about 7,000 deaths in the vaccinated. Those 7,000 deaths, over 90% of them are in individuals over 65. So they're in this higher risk group that really can't get the benefit of vaccines. So the 1 in 500,000 chance of dying is a very broad number, but you can really individualize it. So if this is an individual under the age of 65 who does not have a reason that they wouldn't respond to the vaccine, your chance may even be lower than this.
Sandy, thank you for your contribution. Really appreciate it. One more booster question. Second jab induced a strong inflammatory reaction in me. Have you noticed that the 50 microgram boost induces less?
So I don't think we've had enough experience or enough data, but it does look from the limited amount we have that, yes, there is less reactogenicity with the lower dose. And we'll get more as we go forward. But yes, from the limited data we have, it is less reactogenicity. You
think phenothiazine type drugs could be used in a similar way to Fluvox?
Yeah, I don't know yet, actually. It'll be interesting. So Luvox, just for people, is the brand name for the not so specific the serotonin, selective serotonin reuptake inhibitor. I'm not sure, though, that it's going to be working through the calcium channels. We're actually thinking it's a signaling pathway that at the end is an IL-6 upregulation. So a question again about Fluvoxamine.
Are there any studies debunking the claim that vaccines worsen existing depression and anxiety? A friend's psychiatrist is spreading this misinformation.
Yeah, no, there's no data to suggest that that is true. So it's really unfortunate that that misinformation is being spread.
Thank you, Axis, for your contribution. My friend got shingles 2 weeks after taking the Pfizer vaccine. He's eligible for a booster. Should he take it, the J&J, or just not worry about it? He's immunocompromised.
Okay, so excellent question. We certainly saw an increase in the number of shingles cases post-vaccination. So what we're actually, I think the first question I would want to ask your friend is, have they gotten the shingles vaccine? I would want to get at least one of the shingles shots under their belt, maybe even the second one, right? It can be given 2 months apart before considering that booster, just to reduce that chance. So that's at least what we're doing. If someone got shingles afterwards, we go ahead and we get those shingles vaccines under their belt. These are an excellent vaccine. And then we have this discussion. And as you say, he's immunocompromised. This is someone we probably would talk about getting that booster in.
Sarah, thank you very much for your contribution. And I just want to tell everyone that we're going to wrap it up soon because amazingly it's almost been an hour and Dr. Griffin's time is valuable. A lot of great questions still remaining, unfortunately, but maybe you could convince Dr. Griffin to do another one of these sometime in the future. All right? Let's see. Can you explain without bias why a healthy person should get vaccinated when they can most obviously, according to data, fight it off, not considering the already vaccinated, elderly, and vulnerable?
Yeah, a cruel world. I'm not a big fan of saying you have to get vaccinated for everyone else. I like to look at your specific risks, and let's go through that. Let's say you're young, you're healthy, like many of our people who are asking questions. You might say, I'm not going to die of COVID. I think I'm at low risk of ending up in the hospital. So the big specter for a lot of people is this high incidence of post-acute sequelae of COVID. Not only are we seeing a large number of younger individuals ending up with post-COVID issues, cognitive issues, fatigue issues, but we're also seeing a significant increase, and we talk about this on the COVID update, into non-COVID causes of death and non-COVID issues. Increased heart attacks, increased strokes, increased new diagnosis of diabetes. Getting COVID really wrecks with your system. So it's not just about, am I going to live or die? It's not just about, am I going to end up in the hospital? The amount of ongoing inflammation that can be triggered by an acute COVID infection, we're still not sure what all those long-term negative effects will be.
Lost Laboratory writes, based on data showing stronger immune responses with longer interval between doses, would you recommend parents wait longer than 3 weeks for getting their kids dose 2?
All right, Lost Laboratory. I always use my daughter Eloise as an example. Like, Eloise, once you're in the 90s, it's already an A. You're trying to get an A+. Kids have great immune systems. We're getting to higher antibody levels, which, as I've said, correlate on a community or population level with response. So I'm not sure you need to wait more than 3 weeks. The 3-week interval is to try to get into those 90s without extending that period of time when you have lower protection.
Mary, thank you very much for your contribution to the incubator. Sam, thank you very much for your contribution. Patricia, thank you so much for your contribution. And I would like to thank Dr. Griffin for doing this tonight. I hope you enjoyed it.
I did. Great questions. Thank you, everyone, for showing up.
And I want to thank the moderators for helping out tonight. And I would like to thank everyone for coming. And perhaps we'll do this in the future. If not, of course, you can always tune in next week to the Q&A with A and V. Thanks, everybody, and good night.